Okay, what we're going to do with this video cast is really pick up from the lecture. I suggested to you that it might be advantageous to just go through the chemistry of the system again and then complete the computer-aided molecular design part of the unit. So what we're going to do is really just whiz through the pharmacology, and for those of you who are picking this topic up outside of the lecture course, the hormone physiology and its effects can be read more or less from the PowerPoint slides anyway. Okay then, so let's just go through these things and revise. Remember that the hormones that we're going to be talking about, oxytocin in particular, is derived from the hypothalamus, and we discussed the structure of the hypothalamus and its neurosecretory cells, and the fact that the PVN complex is really important in orchestrating the endocrine responses of these hormones, the autonomic and behavioural responses of these hormones. And we agreed that you'd covered this topic with Dr. Ebenezer, or certainly with the endocrine unit. So we're not going to spend too much time talking about these matters, and there's a link there to a website that goes into a little more detail. Okay. Okay, so from the neurohypophysis, which is the neural lobe or the posterior lobe of the hypothalamus, this is the storage site for oxytocin and vasopressin. We're treating the two, in some respects, we're treating the two peptides in a very similar fashion because they are closely related, and it has some, it impinges on what we're going to discuss about drug development for oxytocin compounds, which is why we're discussing them together. Just to remind you that the axonal release of these neuropeptides comes about by a stimulus on a nerve body, synthesis of a propeptide, which is a transcript which contains the transport protein for the peptide and the peptide itself, and as that protein moves down the nerve under being driven under an action potential, you get axonal transport and processing down the axon until you, in the axon terminal, you get release of the peptide and protein carrier, which are intimately tied to one another. The thought is that the protein carrier is certainly required to deliver the peptide to its receiving nerves, or its to receiving tissues, but it's also to protect the peptide from degradation. It also facilitates reuptake of the vesicles back into that neuron when they're not used. So a reuptake system. Okay, oxytocin and vasopressin are peptide hormones. We mentioned them before. Here's the cyclic structure of oxytocin. And vasopressin is very similar. The differences are here that certain residues are around the core structure. So for instance, here there is an aliphatic side chain residue that's replaced by a phenylalanine residue in vasopressin, but more drastically, and that's a fairly conserved substitution, but more drastically in oxytocin and vasopressin is on the tail of the peptide. This aliphatic hydrophobic residue is replaced by an arginine, a basic residue, and that in some ways delineates the oxytocin vasopressin hormones pretty strongly. So we have cysteine 1, a tyrosine residue, an isoleucine residue in oxytocin, a glutamine residue, asparagine residue, and these are neutral amide side chains, as we mentioned in the lecture course. We have the second cysteine residue, which is going to form the cyclic structure by disulfide bridging between the two cysteine side chains, and this is thought to be important in the activity of the peptide, as you'll see later. We have a proline residue that is now moving on to the tail of the hormone from the cyclic structure, a leucine residue, and then the glycine residue here depicted as GlyNH2, and that's because it's actually a glycinamide. Again, interestingly, it's terminated in an amide residue, and these amides, the glutamine, the asparagine, and the glycinamide residue are interestingly conferring a fairly neutral structure to the molecule, not necessarily highly charged. Whereas in vasopressin we have the arginine, which obviously would confer a distinct positive charge on the hormone. Okay, the cyclic nonapeptide saying that, telling you that there's 9 peptide residues in the whole molecule, can be secreted at central and peripheral sites and acts at a 7-transmembrane GPCR, oxytocin receptor, and it's associated with numerous physiological roles, including the uterine contractions, which is one of the main roles for oxytocin in the formulary. Regulation of sexual response behaviour and emotions is an up-and-coming topic that is hitting the literature at the moment. It's always been there as an underlying effect of oxytocin, but also the emotional response, the more neural function of oxytocin is becoming uppermost in people's interest. Oxytocin is a natural agonist, but this compound is used clinically to induce labour in pregnant women by inducing uterine contractions during the 3rd stage of labour, and also used to control postpartum bleeding or hemorrhage if that happens to occur. So you can see that contractions, muscle contractions, but also controlling blood flow are quite interesting properties of oxytocin. Atosabam is a synthetic oxytocin antagonist, and this compound is used to establish acute treatment to delay the onset of preterm labour if there are issues within delivery there. So we have a natural agonist, oxytocin, which is used, and we have a synthetic oxytocin antagonist, oxytocin-based antagonist, which we're not going to discuss too much, or we're not going to discuss at all in terms of structure, but is there for consideration. Although there are as yet no confirmed oxytocin receptor subtypes, and there's probably unlikely to be so, OT receptors, so there's only one oxytocin receptor. OT receptors show very close similarity to vasopressin receptors, particularly the V1A subclass of receptor, and these, as we've already discussed, these peptides differ by only 2 amino acids, so you would expect some similarities in the receptors. Okay, drug companies are looking into oxytocin receptor agonists for potential treatments in a few areas, and they may not apparently be linked. Abdominal pain. irritable bowel syndrome, and you can see how they may well be a bit related. Autism, well, maybe not so, but autism is a multi-component disease, and there is thought to be a link between the stomach and the brain in autism, which is an interesting observation. Erectile dysfunction, that's quite different again. Lactation, induction and maintenance. Some people have problems maintaining lactation during support of their babies, and it may be a need for these compounds there. Postpartum hemorrhage, as we've discussed. Post-traumatic stress disorder most definitely indicates a neural component, a neural topic that is of interest to the drug companies. Control of pain. These compounds may well have a role in pain that's emerging, and anxiety. So we can see that quite a diverse range of potential treatment areas to be addressed by new compounds. So what we're going to do is just go through the essentials of oxytocin again, like we did in the lecture. Oxytocin can be reduced from its structure to a schematic version, which I would encourage you to use in any discussions of the compound. The absolute chemistry is obviously important, but we can reduce it to a schematic discussion. So we have the cysteine residue at 1, like we mentioned before, tyrosine 2, isoleucine 3, glutamine in 4, asparagine 5, cysteine 6, and that completes the cycle. Proline at 7, leucine at 8, and then the glycinamide, or terminal glycine residue that's turned into an amide at 9, making our nonapeptide. Our nonapeptide has several ways of appearing in the literature. It very rarely appears as that full name, cysteine, tyrosine, isoleucine, and so on. Contracted to the 3-letter codes, yes, it does appear in its contracted form, cis, tyr, al, glin, etc. And then a single letter code representation of the hormone as well, if you're discussing the structures. Here is a computer graphics version of one conformation of oxytocin to highlight the nature of the ring. So we've got a ring in red and a disulfide link that pins the ring together. Then we've got the tail coming in green, just to give you a sort of a sense of the shape of the molecule, and then I've put the residues on in the structure below the backbone version, just to give you a feel for what that hormone looks like. Right, we mentioned that oxytocin has a short lifetime in the body, so as a result, oxytocin has to be carefully controlled. It has to be infused intravenously to maintain its action, because its half-life is so short. And we discussed the reasons for that. There's metabolism, an action called oxytocinase. It isn't necessarily a recognized enzyme. It's an enzyme action that is linked to a zinc-dependent aminopeptidase that's in the body, and this cleaves vasopressin and oxytocin between residues 1 and 2 and 2 and 3. Then there's in the uterus there's a disulfidase action that appears to be enzymatic and does cleave between the cysteine residues to reduce them to cysteines from the disulfide, and hence deactivate the hormone. And then in the uterus again, there's a post-proline endopeptidase that breaks between residues 7 and 8. And in the brain, there appears to be an endopeptidase, again breaking between 7 and 8. And it appears that when these conversions happen, the neuropeptide is deactivated. Where are we with oxytocin? Well, I've mentioned the usual applications for oxytocin in the BNF. When given intravenously or intramuscularly, it doesn't enter the brain significantly, and the drug is destroyed in the GI tract, which is going to give us problems if we think about orally delivering this type of compound. And I'm sure you've discussed that with Dr. van der Merwe and Dr. Waldo. More recently, oxytocin is being employed for actions that are more related to its psychosocial arena, and these are more central than peripheral, and people are in some ways treating oxytocin as a street drug because they feel there's a building popular press that feel that oxytocin does facilitate certain strengthens emotional ties, heightens sexual awareness, and so on. So these less traditional routes that are being used recreationally are happening, and the way in which people are doing this is by using oxytocin sprays nasally. Here's a description of the intranasal administration, just to give you a revision of that pathway, because I know you've done this with Dr. van der Merwe, and certainly some of you have done it in your special topics. So I'm not going to dwell on this too much. Okay, if we want oxytocin to live longer as a peptide drug, then we've got to do things to the structure. And one thing that has happened is the development of a longer-lived version of oxytocin known as carbetocin. And carbetocin's half-life is, you can see, between 30 and 60 minutes, much longer than oxytocin, but still not vastly increased, but certainly of longer duration. And this is because the chemistry around the cyclic part of the structure has been changed. It's mainly the cysteine 1 residue in the ring that's been altered. A removal of the amino group, the terminal amino group in the N-terminus, effectively, of the peptide has been removed, and a sulfur has been eliminated by synthetic chemistry to change the cysteine to a non-standard amino acid residue, and the resultant cyclic peptide is active at the oxytocin receptor, but more importantly is more stable. However, the receptor action is down, and the activity at the vasopressin receptors V1 and V2, there's a myometrial component and a renal component, and V1 and V2 act in those areas. This compound acts as a partial agonist at the receptor, also highlighting the very comparable nature of these compounds, despite the difference in the arginine residue on the vasopressin structure. This means that oxytocin compounds can, if you change their structure, can act at the vasopressin receptor, and we'll discuss that a little bit more later on. Non-peptides. There's a quote here from, I think it's Alan Borthwick, in a recent review. There is a need for non-peptide compounds that are pure agonists and have the potential to be orally bioavailable and more selective against the vasopressin receptor. The advantages will be that these could have potential against the emerging disorders mentioned earlier, and particularly the bioavailability of active compounds against the central targets, where we know oxytocin can't easily reach those targets, other than maybe through the nasal routes. Okay, this is where we get onto the core chemistry that I said that we would go through again and revise. So there are known non-peptide V2 agonists, and this company Ferring, which is a Swiss-based company but has departments in research departments in the UK, in fact pretty locally to Portsmouth, they knew that these heterocyclic compounds, and this particular compound, which we'll call Ferring compound 1, has V2 agonist action, but also is active at the oxytocin receptor. So we show some activity at the oxytocin receptor. And Ferring's wanted to exploit this in development of oxytocin receptor specific compounds. It was also known by publications, patents and intelligence that a Wyeth-Ayerst compound, a Way compound, had some activity against the oxytocin receptor as well, albeit slightly less than Ferring 1. And then also an Otsuka compound had some activity. So these structures indicated that there may be some mileage to build on these chemical structures to pursue oxytocin activity. They all have at least a common framework here, which involves a heterocyclic ring amide connected to an aromatic ring and being extended onto a substituent. So here and here. We can see that also there's an aromatic ring, so you might want to think that pattern is actually extended, but you'll see that there is a reason for just highlighting this bit a little later on. You will also note that this slide is called 1. There's a circle in the right hand corner with a 1 in it. This is approach 1, and we have a second approach 2 that we need to discuss. So Ferring's strategy to develop compounds in a recent patent was to make a library of, they actually made a library of 50,000 or so compounds. 50,000 compounds. In the lecture we mentioned the approach called combinatorial chemistry, which I hope you will pursue on the internet and have a little look about what combinatorial chemistry comprises. But in the discussion, hopefully the idea of combinatorial chemistry will emerge. The technology of combinatorial chemistry, its robotic approach, its machine-based chemistry is another issue for another day. So the Ferring strategy, their hypothesis was that a library designed around known V2 agonists could yield O2 agonists. And in general terms, this sort of highlights the libraries that they put together, although we will see there's deviations from these. But the linkage between the central aromatic carboxylic acid, which was the second structure to appear, and the possibly cyclic amine that's shown in red. So the red colour coding is meant to show the fragments going together. Was one dimension to this library. A second dimension was how can we link another heterocyclic moiety to the amino terminus in this carboxylic acid, this modified aromatic carboxylic acid, and the R group is meant to indicate substituents on the aromatic ring as well, which were investigated. So the coupling together will lead to a series of compounds that is more or less of this structure. This was the predominant nature of the library, and the substituents on the aromatic rings went further than R. Halogens were looked at and other substituents, but this gives the essential feel for the chemistry, and this was covered in BioOrg Med Chem Letter publication, which is given at the bottom in 2004, based around a patent that was put out by Ferring. And there's our if you like, our pharmacophore, or our reappearing motif. I'm not going to spend too much time talking about the high throughput screening because we did that in the lecture. But just to reiterate, just to say very briefly that this is how you would genetic engineering of a mammalian cell type would allow overexpression of either the oxytocin or vasopressin receptor or receptors at the surface of that cell, which allows us to bind compounds, test compounds, all the natural ligand to that receptor. The signaling machinery should be in place in the cell, and that allows a signal to go down to a further level. This further level would be a signal transduction plasmid, which is in the system that's responsive to the signal, and this plasmid allows a light output from a luciferase-luciferin coupling that allows, if the signal goes down by agonism, you turn on the signaling, which is what happens at a GPCR, you would get light output. And of course antagonism would suppress this, partial agonism would give you an incomplete light output, and the intensity of that light is reflective of the potency of the compounds. So we've got our 50,000 library, the compound library. How do we whittle that down to a hit compound? So here's the one of the compounds that came out of that library, ferring compound 27. You could see that the oxytocin activity is now slightly up on the earlier compound, compound 1, at the concentrations tested. It's still not a super compound, but we're starting to see some interesting activity. And you can see that our aromatic amide extended into the red region has already resulted in a heterocyclic compound, or a heterocyclic extension of this system. And in some compounds, this is a common motif in non-peptide compounds that mimic peptide action. Tricyclic heterocycles are quite common through attempts to mimic peptide action. And we did discuss the origins of those compounds when we went through the chemistry within the lecture. Okay, so we've got our red region. Our blue region is an extended, the amines have been attached to our amine on our core structure by urea linkage. And I did tell you in the lecture that to remember the urea chemistry we did in the first year, or the quotes carbonic acid chemistry. Ferring compound 28, you can see that, and this is why the aromatic structure wasn't chosen as part of the motif, because that can change. This can be fairly consistent, a heterocyclic ring amide aromatic motif. Our aromatic ring can change to a heterocyclic aromatic ring. It's giving a little variability there. And so here is our compound now elaborated with amines. In this particular amine, this is an amino acid derived amine from a heavily substituted proline. But we have, look at the activity now. We've got some very interesting activity at the oxytocin receptor now. So this looks a very promising compound. And this was selected for the hit to lead progression. So taking from a hit from the library, we now take that hit and move it to a lead compound that's going to take us towards our drug. This is a mix and matching approach. It's also referred to as scaffold hopping. And the lecture number 9, I believe it is, that I put out as a video cast, is about fragment-based ligand development or design, and talks about scaffold hopping and the reasons for it and the approaches one can take, and integrates very well with what we're discussing here for the oxytocin compounds. So I hope you'll pull the two. You'll take the effort to pull the two topics together, certainly in your mind. But this is to take the opportunity to stress that the fragment-based design lecture is a very important lecture to read. The ferring compound 28. The blue region of the molecule, a further 100 different amides off of this proline were investigated, and also thioamides in addition. And thioamides led to increased oxytocin receptor agonism, but also apparently increased the vasopressin 2 receptor agonism. And this is not desirable, so a strategy had to be taken to try and avoid this problem. So they went back to, let me just go back one, they went back to one of the other compounds that had some activity, and what they did is to mix and match these structures to try and see if the oxytocin receptor action could be retained and the vasopressin 2 agonism could be suppressed. And I'm calling it a mix and match approach. It's called scaffold hopping because you're also taking potentially taking an interesting pharmacophoric scaffold and swapping them around, changing them, hopping from one to another. I've also referred to them as chimeras. A chimera is actually quite an interesting term because a chimera is a match between two different beasts to lead to another beast. So, for instance, what would be the griffin? Look it up online. Is a mixture between two beasts to give a mythical beast. And chimera is also used in biological terms to refer to mixing proteins in their structural characteristics, often by genetic engineering. So this compound, by swapping the head groups round, the head group swap made a very potent oxytocin receptor agonist and decreased substantially the vasopressin 2 agonism. It led also to a compound that, this is ferring compound 39, it also led to a removal of a stereocenter, and stereocenters within molecules are can be problematic, and it conferred selective activity, and this became the candidate compound for ferring's progression towards a drug. So the second approach, and the reason, in some ways, the reason for this podcast, videocast, is to go through and revise structure-based design approaches that can be taken alongside compound development, but also in parallel. Sorry, that does mean alongside. In parallel, because most companies would want to know how these compounds interact at a GPCR receptor. So this is a revision of a potential approach. So structure-based design, this is a requirement to attempt to understand the structure of receptor or binding site and its interaction with an endogenous ligand. And the oxytocin receptor currently has no structure determined for it experimentally, but we have talked about how to progress structure-based design when this is the case. So what can we do? Well, what we can do is we can take the family of GPCRs, and I'm taking this opportunity to show you the family tree, which is why I've called this slide Meet the Family. This is to show you the number of GPCRs that are known within the body to the date of 2011, and their various branches of the family tree. And there are structures for quite a few of these receptors now appearing at a very rapid rate, and understanding of the receptors and their action is expanding almost exponentially. So the oxytocin receptors on the biogenic amine branch of the family tree, highlighted here, and we can take some near coordinates from a relative on that, off of that branch, not an extremely close relative, but close enough. So here, and this is slightly different to the discussion that went on in the previous lectures when we talked about homology modeling before, and we used the oxytocin receptor as an example there. In that example earlier on in the course, we talked about maybe using the adrenergic receptor, which at the time was a known structure, and mimicking the structure or performing homology modeling of the structure to lead to an oxytocin receptor model. Well, we can do the same with adenine A2A receptor, which is actually slightly closer, a slightly closer relative, just to show you that you can use other receptor types as well. And we have X-ray coordinates for the adenine receptor in an active conformation. So this is one course of events for the oxytocin receptor. From the adenine receptor, we can analyze the sequences, do secondary structure analysis, and thread our structural characteristics of our oxytocin receptor sequence onto the adenine A2A receptor. And this oxytocin receptor model is actually fairly nearly ready for docking of resultant complexes, but it's probably quite crude, and we know that that is the case because of the complexity of this protein. So there are some other things we need to do. And we've got to refine the structure. We talked about the fact that you may need to reorient the helices within the receptor model. Just address the twist of the helices as well. They certainly need looking at, and the tilt angle of the helices in the 7-trans membrane bundle of the GPCR. And these are all things we've discussed before, so there's nothing novel here. And the side chains, and certainly including the active site, need to be revisited from that initial model. Do these side chains constitute a good active site for our oxytocin ligand, or indeed agonist compounds for drug development? And we may have to remodel the loops in the structure, because these are specific. These appear to be, the extracellular loops in fact appear to be specific for particular ligands in the family. So we may have to look at those as well. What we can do from our models is we can see which models are fit for purpose, because we can take quantitative structure activity relationship data from our Ferring library, our non-peptide compounds and peptide compounds if we have those as well. We know their structures, or we can mimic their structures by computer-aided molecular design modeling, and we've discussed how we do that, small molecule modeling. And we have quantitative data about their structure and activity against a receptor because of the high throughput screening, and biological data for the compounds. Oops, sorry. And what we can do is try and find the fittest model, the one that fits the QSAR data very well, and progress that to the fittest working model for our oxytocin receptor. Once we've done that, oxytocin, if we are going to look at oxytocin and how it binds at the receptor, oxytocin is actually an incredibly flexible molecule. It's got many rotatable bonds in it. It is known by NMR analysis, spectral analysis, that the compound has mobility. It has certain conformational preferences, and that's a more complete story of what happens with oxytocin. But one of the things we've discussed previously is that it's desirable to try and locate the global minimum for the compound, and even if the global minimum is not the conformation that binds at the receptor, and we've discussed that previously as well, it's most likely a near local minima that would dock at the receptor. So an understanding of oxytocin and its conformational properties needs to be addressed, and to try and locate these global minima and local minima that may be of interest for our structure-based design. One way in which we can do that is to do molecular dynamics, and we discussed molecular dynamics earlier in the course. This is where random motions of the atoms are allowed under thermal conditions, and we look at a parameter that's linked to the geometry of the molecule and see, or the energy of the system, and see how that energy varies with time, and you look for transitions in the structure that lead to significant lowering in the energies, and these energies should be reflective of the global and local minima. We're talking about the oxytocin conformations, and you would have to perform molecular dynamics with this complex system to try and understand its conformational properties, to see if you can't locate a global minimum or near local minimum that would be useful for the docking. The ring is one issue, and the tail, which is actually quite flexible, is another issue. And what actually is found is the ring takes up preferred conformations and the tail takes up a family of conformations due to the flexibility, but is actually quite flexible. It's worth bearing in mind that for some of the non-peptide compounds you would have to perform a similar approach, because they will have considerable number of considerable number of rotatable bonds in them, and may well have complex conformational properties as well. So molecular dynamics would lend themselves nicely to looking at those compounds as well. Once we've found the conformation, one would dock a low energy conformation into the receptor model, and then that would allow, once we've docked that compound, further dynamics, molecular dynamics on the complex would allow, and in effect an induced fitting of the receptor and the ligand to find a more comfortable fit, and then from that you could see how that compound wants to interact with the receptor. You can derive pharmacophoric features for oxytocin at its receptor, its key interactions, and 3-point pharmacophores will allow you to search for further non-peptide compounds within compound libraries. We've talked about grid analysis and molecular field analysis of compounds, so this is putting it into a little more context for our oxytocin compounds, and its facility in searching for other compounds in other libraries. How do we find new compounds? Well, you can look for features. We know, for instance, that we have these consistent features within our compound. You can pre-screen a library, which is, we mentioned SMILE strings, and you could pre-screen a library for those strings, and that will help you tie down filter out compounds that may not be of interest, or you could do that at a 2D library level. But now we have this pharmacophoric information that allows us to search 3D libraries for compounds that will fit into the receptor. We can get fragments as a fragment-based design approach that may be desirable to put within our receptor model, and this fragment-based design approach will allow us to bolt together new pieces that will fit into the receptor and allow us to design new compounds. So this last slide is really pulling together the aspects of fragment-based design, pharmacophoric filtering, and so on. Okay, sorry that's been a little bitty video cast. I had a few interruptions along the way, but I hope you've got the essentials of what I wanted to get over to you with the last area. Try and integrate it with the material that we've had before, and hopefully it allows you to pull all these apparently disparate aspects together in this final case study of how to develop oxytocin, non-peptide based oxytocin compounds with some confidence.